Just like with the T cells, the body contains millions of different types of B cells. Each B cell is capable of responding to a specific antigen. Cytotoxic T cells leave the lymphatic tissue and go seek out and destroy foreign antigens and pathogens, but the B cells stay where they're at. In the presence of a foreign antigen, a specific B cell in a lymph node or in the MALT, the mucosa-associated lymphatic tissue, or in the spleen, becomes activated. Then it undergoes clonal selection, forms a clone of plasma cells and memory cells. So the differentiated B cells become plasma cells that are specialized to secrete antibodies. So the B cells, the plasma cells are the effectors of the B cell clones. They secrete specific antibodies based on the specific antigen that stimulated the proliferation. Okay? Those antibodies then circulate in the lymphatic system and in the blood and reach the site of invasion where the pathogen is. So antigen processing in a B cell occurs when the antigen is taken into a B cell and broken down into smaller little protein fragments. And then, just like we saw with the antigen-presenting cells, the protein fragments of that antigen are going to be combined with a major histocompatibility complex. Then it's moved to the cell membrane of the B cell. Once that is presented in the B cell membrane, the T cells recognize that complex, and they deliver a co-stimulation that's needed for the B cells to be stimulated to differentiate and proliferate. So the helper T cells also are going to produce some interleukins and cytokines that continue that enhancement of stimulating and activating those B cells. Once the B cell once it's activated, it undergoes clonal selection. So that results in forming clones of those B cells, and those clones are going to be plasma cells and memory cells. So the plasma cells are differentiated or specialized cells that will secrete antibodies. So a few days after exposure to a particular antigen, a plasma cell secretes hundreds of millions of antibodies every day for about 4 to 5 days. Then the plasma cell is going to die. Of course, it can't secrete any more antibodies. So the antibodies are going to be traveling in the lymphatic fluids, in the lymph, and in the blood to the site of invasion where those pathogens with those antigens are located. The memory B cells don't secrete antibodies, just like we've seen with the other types of memory cells. They are clones of that specific cell that was activated, so the next time the body comes in contact with that same antigen, they can very quickly proliferate and differentiate into more of the same type of plasma cells, secreting the same types of antibodies. So different antigens stimulate different B cells to develop into plasma cells and those same types of memory cells. So all of the B cells of a particular clone are capable of secreting only one type of antibody, and that is determined by that antigen that stimulated that proliferation. So what are these antibody things we've been talking about? An antibody can combine specifically with the epitope or the antigenic determinant on a specific antigen that triggered its production. So the antibody you can think of sort of like it matches an antigen like a lock and a key. So a specific antibody binds a specific antigen and only that specific antigen. So antigens are glycoproteins, and they belong to a group of glycoproteins called globulins. So for this reason, they're also known as immunoglobulins, globulins of the immune system. Makes sense, right? Antibodies contain 4 polypeptide chains. A part of those chains is called the variable region, and that is where the antigen binding sites are. So the variable region, variable means it can be different, is different for each type of antibody. It's the part of the antibody that recognizes and attaches to a specific antigen. Most antibodies have 2 antigen binding sites. There's also a constant region that's almost the same in all antibodies of the same class. That area is responsible for the type of reaction that occurs. What does the immune system do once that antibody binds that antigen? The structure of that constant region also serves as the basis for distinguishing the 5 different classes of antibodies. We designate those antibodies as IG, IG is immunoglobulin, IgG, IgA, IgM, IgD, IgE. All right, so how do you know what's what? Trust me, you're going to learn much more about this later on. For now, we just need to understand that each class has a distinct chemical structure, and because of that, it has a very specific biological role. So what happens? Antibodies bind antigens, and they can stimulate several different reactions by the immune system. So let's look at some of those actions that can occur. So the 5 classes of immunoglobulins vary in their abundance, they vary in their structure, but what they do is based on that structure and the actions they provoke in the immune system. So they can simply bind to an antigen and block or neutralize some of the bacterial toxins or prevent attachment of some viruses to body cells. If the antibodies form against antigens that may be on cilia or flagella of some of the bacteria that are very mobile in the body, then that antibody-antigen reaction may cause the bacteria to lose the ability to move throughout the body, and therefore the infection can't spread to other body tissues. Antibodies have 2 or more binding sites, right? So that means they can bind to more than one antigen. So the antigen-antibody reaction may cause cross-links between pathogens, okay? So that can lead to agglutination, just like you saw with agglutination when you did blood typing. So phagocytic cells ingest agglutinated microbes more easily. So it's sort of like it may in some way tag those cells and alert the phagocytes to come in and to digest away those. Also, some of those complexes will become very heavy in the body fluids, and therefore they will precipitate or fall out of that solution. And again, that's going to allow the immune system to respond, those phagocytes to come in and destroy those complexes. I mentioned already that it can enhance phagocytosis by attracting the phagocytes to come in. So the antibodies enhance the activity of the phagocytes, but they can also activate a complex system called complement. So antigen-antibody complexes initiate the pathway of the complement system. So the complement system is a system made up of 20 to 30 proteins that are produced by the liver and found circulating in the blood plasma and also in certain tissues in the body. So the complement proteins destroy microbes by causing phagocytosis or cytolysis or enhancing the inflammatory response. They also help to prevent excess damage to tissues. So the complement system acts as a cascade, very much like a domino effect, where one reaction triggers another reaction, which then in turn triggers another reaction, which triggers another reaction. So you get the picture here, right? So each succeeding reaction, more and more and more of whatever is causing that reaction is produced. So if it's causing phagocytes to come in, then each reaction produces even more chemicals that cause even more phagocytes to come in, or even more precipitation occurs. So whatever the action is enhanced by the complement system. Immunological memory is due to the presence of long-lasting antibodies and lymphocytes that have a very long life. So remember, these arise during the clonal selection of those antigen-stimulated B and T cells. Immune responses, whether they're immune or whether they're antibody-mediated or cell-mediated, happen much more fast, so much quicker and much more intense after a second exposure or even subsequent exposures after that to a particular antigen after that first response. So during that first response, the response to a particular antigen, only a few cells have the correct specificity to respond. So the immune response can take several days to build up to maximal intensity. Because thousands of memory cells will exist, though, after that first encounter with that antigen, the next time the same antigen appears, those memory cells can proliferate and differentiate into helper T cells, cytotoxic T cells, or plasma cells really quickly within hours. So one way to measure the immunological memory is to do an antibody titer, which is just how many antibodies are in the body serum, the plasma serum. So after an initial contact with an antigen, no antibodies are present for several days, but it does slowly rise, so the antibody titer occurs and fluid levels of antibodies rise. So this is followed again by a gradual decline in the antibody titer, and that's what we call the primary response. So the immune system, the B cells, have to have time to create a number of antibodies. Those antibodies then are found in the blood plasma. So it takes a while for those levels to go up. Then after there's no more antigens because the pathogen has been destroyed, right? The antibodies stop being produced, so the levels go down. But memory cells can remain for decades. So every new encounter with that same antigen is going to result in an even more rapid proliferation of memory cells. So this accelerated and more intense response is called the secondary response. Antibodies produced during a secondary response have an even higher affinity or attraction for that antigen than the ones that were produced during the primary response. So they're even more successful in getting rid of that pathogen, getting rid of those antigens. So primary and secondary responses occur during microbial infections. Sometimes, though, you recover from an infection without taking any antibiotics or any type of antimicrobial drugs, and that's usually because of the primary response. But if that microbe affects you later, the secondary response could be so fast that the microbes are destroyed before you even get sick, so you don't exhibit any of those signs of the disease. Immunological memory provides the basis for immunization by vaccination against certain diseases, for example, polio, chickenpox. So when you receive a vaccination, usually you're receiving an attenuated or weakened or killed microbe or portions of that microbe, that bacteria or that virus. But they still possess those antigens, right? So your B cells and your T cells are still going to be activated, but they're weakened. The microbes are weakened to the point that they don't cause you illness. So you've activated your B cells, you've activated your T cells because of that vaccination that contained the microbes. So you've made immune system cells, you've made memory cells. So later, when you encounter the living pathogen as a microbe that can infect your body has already produced those memory cells and can initiate that secondary response. So we look at a couple of different ways to acquire this type of adaptive immunity. So naturally acquired humoral, remember humoral means of the body fluids, so naturally acquired humoral immunity results from acquiring an infection or a disease. So if you get the chickenpox, you develop those memory cells, and therefore next time you come in contact with the chickenpox virus, you have a naturally acquired immunity. Artificially acquired immunity is when we get those vaccinations. So we prime the immune system to develop those antibodies. So naturally acquired active immunity, active, remember, means that you received or you got the disease. Naturally means you got it without getting an injection. So when you get the chickenpox and you develop those antibodies, that was naturally acquired, you got the disease. Active because your body created those immune cells. Artificially, as I mentioned, is when you stimulate the immune system to create those antibodies by giving those microbes, weakened versions of those microbes. But it's still active immunity because your body actively created those memory cells. So this is when you get those antigens that are going to be induced during vaccination. So this is, for instance, when you get some of the vaccinations for hepatitis or smallpox or measles. So you're not getting the disease and causing you to create the antibodies and causing you to create the memory cells. You're getting those weakened particles, those antigens that are causing you to. All right, passive humoral immunity. What does passive mean? Passive is not active, right? So this is when your immune system is not making those memory cells, not making those antibodies. So you're getting those antibodies from another source, either through an injection or from a donor. So the B cells are not activated. You're not making memory cells. And the protection from this type of immunity ends when those antibodies are degraded or broken down. So naturally occurring passive immunity, remember, only happens between a fetus and the mother. So when the fetus gets that passive immunity from mother with some of the antibodies that cross the placenta and also in the breast milk. So this lasts for several months after birth and allows the immune system of the infant to begin functioning and creating their own memory cells and their own antibodies. Artificially acquired passive immunity is when you're given injections of antibodies, such as what happens after you have been exposed to the hepatitis virus, also in response to some snake bites and to rabies and some types of botulism. So you're given antibodies. These will last in the body for several